our chapter, I think, had also both the support from Optica and SPIE, so they're not actually, they're happy to co-support.
Okay.
Yeah.
So good afternoon, everyone. Welcome to our online regular Optica seminar series. Today we have Professor Giorgio Volpe from University College London as our today's speaker. Before the talk starts, let me introduce our speaker to the audience. So Professor Volpe achieved his PhD degree in photonics from ICFO, the Institute of Photonic Sciences, Barcelona, Spain, in 2012. During his PhD, he mainly worked on optimizing light-matter interaction with very small quantities of matter down to the single molecular level. After his PhD, he held a 2-year junior position at the Langevin Institute, ESPCI, Paris, France. During that time, Dr. Volpe's research focus shifted to light scattering in complex media and its application to nanophotonics, optical trapping, and imaging through opaque materials such as biological tissue. He then joined UCL as a lecturer in 2014 and currently serving there as a professor. So he works on a vast area of research relying on forefront photonics and fabrication methods. His current research interests are the experimental and numerical study of far from equilibrium behavior emerging in active soft matter systems that are of either biological and artificial origin, such as motile bacteria and artificial microspheres, respectively. With this brief introduction, I would like to request Professor Volpe to kindly take over.
Thank you very much for the very kind introduction. It's very, I guess you started very well my CV, and I was impressed. Thank you. And I guess what I'm gonna do today is, I don't want this to be very formal. So if you guys have any questions, just feel free to interrupt me anytime and ask while I'm presenting. It's not a problem at all. I'm going to start just showing you some examples of systems from different scales. And here there is a video in which you can see some pneumatics that are being rearranging, and they're creating these very mesmerizing patterns when in a magnetic field. If we increase to the higher length scales, what you see here in this video, although things are moving, they're moving very slowly, we have some Janus particles that are the dark colloids that are moving in a background of passive particles. If we increase the scale, what you see here, apologize for the background noise, is some robot hoover, the white disks that are interacting with the boxes that were shifting. And as you can see, by effect of these interactions, there are some patterns that are emerging because the white hoovers, they are reorganizing the boxes and they are clustering them together. And finally, I'm going to stop this. And if you move at even bigger scale, our scale, what you see here, if I can play the video, no, doesn't seem to want to be playing. Well, what you will see here, this is one of the past Boston marathons, and you can see the crowd of people here that are at the start line, and they are, once the marathon starts, they're all running together, and immediately you would be able to see, if the video was playing, some patterns emerging in the group of people. Now, all of these systems, although they span across completely different length scales, what they have in common is that all examples of active matter system, from the molecular scales up to our macroscopic length scale. What I'm going to do today is to first explain what active matter is. I know you guys probably already know this, but just to make sure that there is we're all on the same page, I'll give my interpretation of what active matter is. And after that, I will discuss two main examples of emerging collective behaviors that we observed in our lab that are not trivial, and they are due to the interaction of these active particle, active matter systems with passive particles in their background. But let's go one step at a time. So what is active matter? As you see from the previous examples, in the initial slide, active matter system can be either biological or artificial systems where the individual units can take up energy from their environment and use it to perform autonomous tasks. For example, they can self-propel, or they can create more complex patterns. We can understand active matter better by comparing it against what we know best from a university, which is passive matter. So matter that is either at thermodynamic equilibrium or near thermodynamic equilibrium. In passive matter, what you have is a series of passive units. For example, if we think about colloidal particles, microscopic particles, we can think about just standard Brownian colloids. And these units can be driven to self-assemble to form patterns. For example, here you see a photonic crystal made from tiny, in the order of 1 micron, colloidal particles. What happens once you have systems that are out of equilibrium and they can exploit energy that is available to them in their environment, so active matter systems? In this case, you will typically have a combination and mixture of active units. For example, if you think about living active matter, the active units can be the biological cells that form tissue, plus you can have other units that are not active, for example, minerals or molecules. And in the case of active matter, again, taking tissue as an example, these active units, the cells, and the passive units can take energy that in the case of living tissue is provided by food, so it's a chemical energy, to form complex structures. And I found that the beautiful example of this is bone, because in bone, osteoclasts and osteoblasts, the cells, are continuously playing a tug-of-war game to remodel the mineral matrix of the bone to adapt to our ever-changing weight and level of activity during our lifetime. So if we compare these two mechanisms, the one at the top for passive matter, so matter that is near thermodynamic equilibrium, and the one at the bottom for active matter, so matter that is far from equilibrium, what you will find is usually these passive matter systems tend to be slow, very much static, and not reconfigurable, which is completely different from what happens for active matter system, where you have behaviors that tend to be much more adaptive, dynamic, and reconfigurable. So why do we want to study active matter? Well, there are some fundamental reasons. If we think about living active matter system, we have collections of insects, school of fish, flock of birds, herds of mammals, and as I tried to show in the first slide, even human crowds, for example, during the Boston Marathon. Understanding the patterns that emerge in this sort of system is a fundamental scientific challenge for 21st century physics. There is no yet a theory that describes what happens when a system is far from equilibrium. But active matter has also found and is finding more and more applications. So there are also technological reasons why we want to study these systems. If you think about 10, 15 years ago, most of the applications for active matter, especially for microscopic active matter, were at the crossroad between biomedical applications and environmental applications. And people, researchers, were thinking that by combining the possibility of transporting actively cargoes, manipulating them, or sensing your environment, you could develop a plethora of behaviors that could be potentially useful for this sort of either bioremediation task or biomedical drug delivery applications. What happened in the last 10 years is that active matter systems actually started to see a spread of possible applications that they can be used for. And one of these possible applications is to develop materials that are out of equilibrium, And as I shown you the previous slide, this means that we can possibly create artificial materials that are closer to what living tissue does, rather than being stuck with the rules of classical thermodynamics. But of course, active matter systems are much broader. For example, they can also include the behavior of groups, swarm of drones, and robots, which are intensively used nowadays for several applications in search and rescue, in surveillance, but also for agricultural and forest monitoring applications, because obviously the area you can cover using several drones is much more efficient than what, and it's a big help to what human can do alone. The main point, however, is whichever application you have in mind for active matter system, or whichever fundamental behavior you want to understand, this active matter system typically develop in complex environments where there is always crowding and complexity in the terms of physical barriers. And if you think about bioremediation, that, for example, will happen in soil, which is a porous material. If you think about drug delivery, that will happen in tissue, and there is a complex network of channels and vessels. Or even on lab-based situations, you will have to navigate through lab-on-a-chip devices where there are channels, flows, and so on. So what I'm going to show you today is two different results from our lab, where we have shown some interesting behaviors that emerge in the interplay between active particles and passive particles. Now, these behaviors that I'm showing are highly nonlinear, and they are based on a feedback being developed between the active particles and the passive particles. So the actual active matter system and the background, the complicated environment they live in. So I'm going to show you two examples. The first one is stigmergy of active colloids, and the second one is more towards application, and is the case of a programmable colloidal laser. Let's start with the stigmergy of active colloids. Now, I know the stigmergy is not a word that maybe a lot of us have heard before. So just to define it, stigmergy is a form of indirect coordination that happens as a mean. It develops through communication that is mediated by the environment. So it's not direct communication between the players, it's indirect, and is usually mediated by the environment. I'll give you a couple examples to understand this better. You're probably familiar with the behavior of ants and termites. These are social insects, and the way in which they communicate with each other is to leave pheromones, traces, that other ants can pick up and follow. And therefore you see complex behavior emergence like lane formation, but some species of ants can do pretty astonishing things. For example, they can create rafts, and they can create bridges that help them move through their environment. That's probably one of the most classical examples we can think of stigmergy in the natural environment. But there are actually other situations that you probably experience in your everyday life that are usually not associated with stigmergy, but are actually also stigmergy behavior. A typical example of this is the trails, the paths that are formed by people or animals that are walking on low grass. If you are walking on low grass, because you are walking on it, you end up squashing the grass, and you end up forming these trails, which are not immutable. They can change. But what happens is that when another player, another person, another animal comes in, instead of creating a new path in the low grass, the tendency will be to follow the previously established path. And this is also an example of stigmergy. There is no direct coordination between the players. There is no communication, because a person can have passed through this path hours before. However, there is still this trace left in the environment that a new agent can pick up and follow. Another example of stigmergy is inspired by, again, social insects. In particular, these are termite-like robots which can build complex structures by following very simple rules. And this rule is very similar to what termites do when they build their homes, their home mounds. Termites, when they build their nest, they will start depositing some material, and that will become a queue, indirect queue for other termites to keep on building around that material that has been deposited. And without any knowledge of the bigger plant, they can manage to create very complex nests with very complex network of channels. These are robots that were developed a few years ago, and I think the work was published in a science paper where these robots, following similar rules to the one of termites, can build pretty much 3D constructions. So I'm gonna now talk about our work with active colloids, but before going to the detail, let me actually acknowledge the people that have done the work. So the experiments that I will present were done by Manish Trivedi, who was a PhD student in my group, and the theory and the simulations were performed by collaborators at University of Lisbon, in particular, Professor Nuno Araujo and Dr. Cristóbal Dias. I'm gonna play a video now, and what you see at the moment is two types of particles. The one that look slightly darker, like this half moon shape, these are Janus colloids. They're colloids that are half coated with, in our case, with carbon, and they can self-propel when they absorb light. And then you can see a background of passive particle in which these Janus particles are moving. And you can see the trajectory that these Janus particles are creating their environment. Now, can you see anything that is happening here? I know you probably don't want to answer directly because this is being recorded, but if anybody is brave enough, I'm gonna play it again. Can you see anything emerging here? I'm gonna give you a hint. Look in these areas, and particularly look at these two particles.
Particles are tracing the same path.
Exactly. So what's actually, I'm gonna play it again because I think it's nice to see. So you see this first particle is digging a path through the background of passive, and this second particle comes in and actually quite smoothly follows that path without having to dig its own, most likely. And this is not the only case it's happening. You can see at the edge, I'm gonna play this again. This particle here is digging a path, then the interaction with the passive colloids makes it turn on itself, and actually it ends up reusing its own path that it created before. So there is this behavior, which is not very different in a sense from the traits that are left by humans or animals when they're walking on low grass. Particles are generating a path, which is transient, is not immutable, but if this path doesn't disappear because of the motion of the surrounding Brownian colloids, it can be reused by other Janus particles. And we think this is an example of stigmergy. Now, how does our system actually work? To understand the physics behind it, we need to think about what is propelling our colloidal particles. As I said, we are working with Janus particles that are microscopic colloids. They're half-coated with a material. In our case, is carbon, but it could be gold, could be something else that can absorb light. And they are put in a critical mixtures of water and lutein. Now, water, we all know what it is. Lutein is more like a oily substance. The nice thing about these mixtures is that the water and lutein, below a critical temperature that is around 33, 34 degrees Celsius, are actually completely mixed together, so they form one phase. But as soon as the critical mixture crosses the critical temperature, there is a phase separation between a water-rich phase and a lutein-rich phase. Now, what we do is we exploit this behavior, this critical demixing, to power our Janus colloids. As I said, if you remember, our particles are half-coated with a light-absorbing material. It is carbon for us. And once you shine light at them, the carbon will absorb light and you generate local heating. This local heating is enough to bring the critical mixtures around the particle above the critical threshold, so above the critical temperature, and we can see a local demixing in a water-rich and in a lutein-rich phase just around the Janus particle. Now, this forms droplets just around the Janus particle that can self-propel it because of body forces, pretty much. Now, if you think about our previous system, we have a lot of the Janus particles that are moving in a critical mixtures in a background of passive colloids. If you look at the individual motion of each Janus particles, what you tend to see is that these particles are ballistic, self-propelling in the absence of any obstacles. So this is the first mean square displacement, which you see here at the top of the graph for no density of passive obstacles. Now, as a reminder, the mean square displacement is a statistical quantity, and it quantifies how far on average a particle has moved from its initial position. So in a sense, it quantifies how far in time a particle can realistically go on average. What happens once you increase the density of the passive colloids is that, as you would expect, the motion of the Janus particle becomes more and more hampered. And you can see this because the mean square displacement is decreasing with the density of passive colloids. And this makes sense because the particles now have to push against the background of passive colloids in order to be able to move forward. Their motion is not as easy as it used to be when the environment was fully homogeneous. However, if you remember, we also saw the formation of this transient path. You can see here another example where you have these three Janus colloids that are highlighted with colors, cyan, yellow, and magenta, pink. You can see that the cyan colloid at the front is the one that is creating a path for the other particles to follow. And over time, because obviously the particle at the front is the one that is more hindered in terms of its motion by the passive colloids, the particles at the back can catch up with it and they start to form groups. So there is something counterintuitive here. We've seen that although individual colloids are hindered in their motion, the mean square displacement decreases because of the presence of the passive colloids. Therefore, we should expect that the chances of them to meet will decrease too because they're struggling to move. What we actually see is that this is not the case. As a function of the density of passive particle or the background of passive particle, what we observe is that the size of the groups that are formed is actually larger at intermediate value of density of passive particles. So if you have a homogeneous environment, the groups that will form are smaller than if you have a mildly crowded environment. And of course, only once we increase the density of the passive background beyond a certain threshold, then we start to observe what we would expect intuitively is that the formation of groups is hindered. And therefore, the size of the groups starts to go down towards zero again. But this behavior, where we observe an increase, a boost in the formation of groups and their size at intermediate packing fraction, intermediate density of the passive obstacles, is not trivial, is not intuitive, for the same reason that we mentioned before. The motion of the active particle is hindered by the obstacles. However, we need also to weigh the importance of these transient paths that are formed and that can be reused by other Janus particles. And this is the effect that is dominating at these intermediate scales. How do we explain it from a physical point of view? Well, if you remember, I told you that our Janus particles are moving in a mixtures of water lutein. And once the temperature is above a critical temperature, there is local demixing in a water-rich and a lutein-rich phase. If the particle approaches an obstacle to its side, the demixing at that side of the Janus particle will be hindered by the presence of the obstacles. So the demixing around the direction of propagation of the particle is now not symmetric anymore. it will be stronger on this side than it is on this side. And because of this asymmetrical demixing, the particle will tend to rotate towards the area where the demixing is higher. So there is an aligning torque that acts on the Janus particle because of the asymmetrical demixing caused by the obstacles. And you can see this clearly in these two time frames. The particle approaches this set of obstacles, having them mainly on their left side, and this turns the particle clockwise. Then here, it approaches these two obstacles pretty much head-on, therefore there is no asymmetric demixing anymore, and what the particle does is to burrow through them. But look here, there is a path, and there are some more obstacles there. What happens once the particle approaches this set of obstacles? Well, again, it experiences a torque that aligns it to the path that was present here. So this complex interaction between the passive particles and the active particles allows them to align and reuse these transient paths. And in fact, what we notice experimentally is that the path revival lifetime, so this quantity here, tau, as a function of the density of passive particle, this is something we define as the path revival lifetime, which is pretty much the probability, the likelihood that the area that has been visited by a Janus particle will be visited by another Janus particle within a time lag. The shorter this value of tau, the faster particle will be revisiting previously visited path by other Janus particle. Therefore, the shorter this value of tau, and the more likely is that the particle will reuse an open transient path. And what you see here, the red curve are the experimental data, and the black and gray curves are actually simulations. You can see that if we include this torque, we can reproduce the experimental behavior, but as soon as we kill the torque, then what you observe is that the path revival lifetime increases with the density of passive obstacles, which makes sense because in principle, if your motion is hindered by the presence of obstacles, then it will take you longer to visit a region that was previously visited by an active particle. So this is the more intuitive picture, while the less intuitive picture can only be explained due to this complex interaction with the passive colloids that induces a torque on the motion of the active particles. And in fact, in simulation, if we include this torque in the motion of the active particle, you can see that we can regenerate the fact that the group that form have a peak around the intermediate value of passive obstacles, while if you remove the torque, there is almost no relevance on group formation as a function of the density of passive particle. Actually, if you notice, it's more likely the groups will form a low density than it is at the high densities, which also makes sense because without the torque, then particle can meet more, it's more probable the particle will meet in the absence of something hindering their motion rather than in the presence of obstacles. Only the torque can explain the non-monotonic dynamics we see in experiments. Now, because of this torque and because of this path reuse, what's happening is that pretty much the Janus colloids are leaving trails in the background of passive particles, creating a field memory that other particles can exploit to converge to a population behavior, in our case, the formation of groups. And what's interesting is that if we look at the aggregation kinetics, so how these groups form, we can see the importance of having this torque. Now, the aggregation kinetics, we can model them quite easily using rate equations. C1 here is the rate of formation of monomers, so individual particles. And in our model, you can see that monomers disappear, there is a minus sign, either because a dimer is formed, C1 to the square means that the dimer is formed, so this is the situation here, and there is a rate coefficient that multiplies this process, or monomers can disappear from the system because they join an existing group, the second situation here, again, with a rate coefficient. And then there is another dynamic that is the one of group formation. The number of groups changes in time, either because there is a new group forming, a dimer is formed, that's the first term in this equation, or it can change because, as the first situation depicted here, two groups that are already existing merge into one, therefore the number of total groups decreases. If there was no correlation, no field and memory in the environment, you would expect this dynamic to not show a strong dependence on the environmental properties. However, you can see that with the presence of the torque, we can calculate these rate coefficients, and we can see again that there is the emergence of this peak around intermediate values of the passive background, so the density of passive obstacles. Now, what is interesting here to see is that the driving mechanism for this group formation is not the formation of a new group, because two monomers form a dimer, which is the dark purple line here, but actually the mechanism that is driving the increase in size of the groups is either the addition of a monomer to an existing group, which is the second line here, the one that I'm tracing, or the fact that two groups merge into one, which is the fainter purple line. What does this mean is pretty much, rather than formation of new groups, what is driving what we observe in the experiments is the fact that an existing group will catalyze its growth rather than more groups will form. If you remove the torque, and this is done in simulations, you see that the trend changes completely. There is no peak emerging at low packing, at intermediate values of the density of the passive phase. And what you observe is actually that all of these coefficients decrease as a function of the density of passive particle. That is more intuitive, because the more dense the environment is, the more challenging it is for active particles to move through it and find other active particles and form a group. So this is the more intuitive picture. This is the less intuitive pictures that actually is the one we observe in the experiments. And the explanation for this is indeed the emergence of this field memory, this stigmergy between the active colloids. So I'm going to switch to a different example now, but I just wanted to ask if there is any question on this part that you want to ask already, or we can leave them to the end.
So, one thing, like why specific like carbon coating you have used, like, and what is your illumination condition? Is it white light?
We use a green laser, so 532 nanometers for illuminating the Janus particles, but in principle, any wavelength will do, because carbon is black, so it absorbs any wavelength of visible light.
Okay.
If you were using a different absorbing material like a metal, gold, then you will need to work at the resonance of that metal, which in the case of gold is 530. For silver will be more towards the blue and so on.
And you have also shown like the tracing of the path by Janus particles. So because of the like phase transition between phase two separate phases, so is it reversible process, or like, because when temperature is increasing, then it is being phase transition is occurring, and when the particle is passed through, then will it again be back to the previous situation, or it will remain the phase transition?
Yes, so just to check, I'm understanding your question. You're asking whether the phase transition in the water-lutein is reversible or not,
Yes,
correct?
yes.
It's fully reversible. So if you, what we do is to work in a condition where we are just below the critical temperature and the mixture is in one phase. Once the Janus particle passes an area, because of the local heating, you push the system just above the critical temperature and you get the mixing. But as soon as the particle has moved away, the local temperature will decrease again below the critical temperature and the system mixes again and becomes one phase. So it's fully reversible.
Okay, thank you.
Which is actually part of the reason why this mechanism is so nice to work with. It's very much controllable and there's a lot of advantages. And the level of intensity of light that you need to propel the particle is really low. We're talking about microwatt per micrometer square. So it's relatively low.
So Giorgio,
yeah.
So if instead of having this passive particle to create sort of a barrier, if one uses a speckle field, would the observation be similar?
Depends on. So we actually have. There will be something coming out soon on that, but in part, yes. So what we have seen with speckles is that particles tend to follow the channels created by the speckles, so the low intensity areas. But in terms of group formation, we did not observe the same dynamics. And the reason for that is that although the reason why the particle follow the speckle low intensity area is still due to a torque, but the mechanism behind it is different. Therefore, it affects the formation of groups in a slightly different way. So I would say at the individual particle level is very similar, but in terms of what you end up observing collectively is not. Okay. And yeah, we're probably gonna put the paper on archive on that soon, like in the next couple of weeks. I'm happy to share it as soon as it's available. But actually, what we observe is that speckles tend to promote smaller groups and smaller and more stable groups rather than larger groups.
Okay. Okay.
So, Giorgio, I have a question.
Hi, Pavan.
Yeah, hi. So this is related to the effect of polarization. So, is there a possibility that polarization can play some role in this kind of observation?
You mean the polarization of light?
Yeah, the polarization of light. Yeah.
With the one I presented.
Yes, yeah, the one which you presented.
No, because it's purely light absorption. It's not really polarization dependent.
Okay.
In our case, I mean, it's carbon. It pretty much will absorb anything.
Yeah, yeah.
So
no,
I mean,
because
yeah.
The moment you have multiple particles, sometimes it might actually lead to some interesting collective effects.
That could be possible, yes. I mean, for the geometry we work with, we didn't observe any polarization effect really.
Okay.
So, but I think you're probably right, especially I guess if you're working with metals rather than carbon, where they can be more sensitive, like because of plasmonic resonances, polarization of light. I guess you probably might end up seeing some effect. Now, in the past we were using gold instead of carbon, and we didn't see any major polarization effect, but you also have to keep in mind that our particles are quite chunky, we're talking about 5 microns.
Yeah.
If you were working on smaller scales, I guess it's an interesting question.
Because in some of the iron coated particles, sometimes we do observe some slight polarization effects. But this is I understand the point what you're making in this particular context. Maybe that is
So so you do observe with the iron oxide.
Iron oxide kind of induced, or rather particles which have iron oxide as part of the composition, not only on the surface. So they do so, especially while trapping, they do show some polarization effect per se, but not as pronounced. It's not a very pronounced effect. Yeah.
Okay. So okay, I will be interested to have a chat about that.
Yeah,
but,
yeah, sure.
But I mean, in our case, we didn't have any appreciable polarization.
Polarization. Okay. Thanks.
Shall I go to the next topic? Yeah. Okay.
Yes, please.
The second thing I want to show is still a complex interaction between active particle and passive particles. Pavan have already seen this, so I apologize for the repetition. But I guess we all know in this audience what a laser is. And we know that how things have really, for lasers, have really became a very important part of daily life. You know, it's not just that in education and research we use lasers a lot. You typically, if you're flying somewhere, you will at some point scan your boarding pass on a laser device. And often also a lot of the checkout areas in supermarkets now rely on lasers. So it's really a technology that has become so intrinsically connected to our way of living that we don't even realize anymore that they are all around us. Now, the lasers we're typically used to work with are known as conventional lasers. In conventional lasers, you have a cavity, two mirrors, in which there is a gain medium, and light gets amplified by stimulated emission or radiation. That's what laser stands for. Because of the two mirrors of the cavity bounce the light back and forth through the gain medium, and this gives a chance for, if some conditions are met, for light to get amplified. Because of the cavity, the modes that typically survive these conventional lasers is a single mode. Most likely will be a TM00, which has a typical Gaussian profile. In the last 15 years, 20 years, there has been more interest in developing lasers that are non-conventional, so they don't fit this conventional category of lasing. And a very popular example is the one of random laser, where we still had a gain medium, but now the amplification is not due to light being bounced forth in a cavity, but is due to light being scattered by strongly scattering elements multiple times so that it gets trapped in the medium to give rise to amplification. And of course, because of the random nature of multiple scattering, even the modes that survive in this laser are looking random. Here you have a localized mode, can be localized in time and space, or you can have more diffusive mode that spread all over the structures. But in both cases, they look random, right? All of these lasers, whether you think about conventional lasing or non-conventional lasing, are usually static and are reconfigurable. What we wanted to achieve is a reconfigurable programmable laser. Again, let me first acknowledge the people that have done this work. This work was again carried by Manish during his PhD in collaboration with the group of Riccardo Sapienza at Imperial College London, in particular with Dhruv and Kit, a postdoc and a PhD student in Riccardo's group. So the kind of laser we want to realize somehow mimics the assembly mechanisms that are shown to you at the beginning for living materials. In these living materials, just as a recap, we have active particles, which usually are cells, and passive particles can be the minerals in the bone system, for example. And in the presence of energy, again, that for living material, this is provided by food usually, so it's chemical in origin, can give rise to structures like tissue that can remodel, reconfigure over time. What we wanted to do is to reproduce this self-assembly strategy by mixing active components, the Janus particle, and passive components. For us were highly scattering colloidal particles made of titania, which in the presence of energy provided externally, in our case is light again, they can give rise to reconfigurable structures, clusters of colloidal particles that, if placed in a gain medium, can give rise to lasing, and we can probe the lasing by probing the local spectral properties of the cluster that is being formed. Let's go one step at a time, and let's think about what are the key ingredients we need to achieve our laser. We need three main ingredients. We need an active colloid, which is the one that can drive the whole system out of equilibrium, therefore provide the trigger for work to be done. We need a gain, because without gain, you don't have any amplification. And we need scattering medium that is capable of trapping the light long enough in this device for it to be amplified by the gain. For active colloids, we use, again, Janus particles, which we've seen what they are, and we know that they absorb light, and because they absorb light, they can perform tasks out of equilibrium. Now, I know this question is coming, so I'm just going to answer it already. You don't need a Janus particle to do 90% of the things that I'm going to show you. Any light-absorbing particle or light-absorbing configuration will probably do. We use Janus particle because this is what we know best. And also because some of the things that I will show towards the end of this part of the talk depends on the fact that this Janus particle can self-propel. So in terms of gain, then what we use is an ethanol solution of rhodamine dyes. Why rhodamine? Because it's a very well-established laser dye, and the property, both absorption and emission, are very well known. It's very easy to control. And then as a scattering medium, again, we use microscopic particles that were made of titania. Titania, we chose it because it has a high refractive index. I think it's 2.4, around 2.4. Therefore, highly scattering properties. And moreover, we chose them to be of a size comparable with the size of visible light to maximize the light-matter interaction of interest, that in our case were the scattering ones. So now that we have our three ingredients, we can put them together. We can create our sample. Our sample is a microfluidic chip, where you can see here, one Janus particle is the big chunky particle at the center, and the titania smaller particle that are jiggling around because of Brownian motion. If we illuminate the Janus particle with an external energy source, again, provided by light in our case, what we observe is that the colloidal titania particle that were first jiggling around because of Brownian motion converge and form a random cluster around the Janus particle. And as soon as we remove the external illumination for the Janus particles, so the energy source, what we observe is that the titania colloids disperse again. Now, what's happening is, you guys know it better than I do, and this was already explained by work from Frank Sichos in Leipzig. What we had are thermosmotic flows that are forming in the presence of a temperature gradient near a solid substrate. Now, they're called thermosmosis flow in analogy to what happens for osmotic pressure. In an osmotic system, if you have two regions of a sample that are separated by a semi-permeable membrane, which is permeable to the solvent but not to the solute, and you have a region with higher solid concentration than the other, then because the solid particles cannot cross the membrane to compensate for the difference in concentration, what happens is that the liquid will flow in the opposite direction to dilute the more dense region. Now, with thermosmosis, conceptually something similar that is happening. There is no semi-permeable membrane as such, but the solid is somehow acting in a similar way by creating an enthalpy gradient from the bulk towards the solid. Because there is this enthalpy gradient, the mechanism that drive the mixing of the hot and the cold region is not the diffusion of temperature mainly, of heat, but is a flow that gets established near the solid substrate that goes from the cold to the hot region. And this flow depends on the gradient of temperature and on the motility of the system. And what is nice is that basically this explains what's happening in our experiment. If we are illuminating the Janus particle, we have local increase in the temperature, and this creates a temperature gradient that goes from the Janus particle towards the outside, the outskirts of the sample. And this drives the motion of any titanium particle that are surfing these thermosmotic flows. And in fact, you can see that with a simple model that takes into account the diffusion of the Brownian particles and these thermosmotic flows, we can reproduce our experimental results quite well. The shaded areas here are the experiments. The dashed black line is this model. And you can see how well we can reproduce that initial accumulation phase, but also the dispersal and the accumulation, because this process can be repeated cyclically. So now that we know how we can make our cluster of colloids reconfigurable, and we know that is an again medium, what we wanted to show is that indeed it is lazy. And for doing that, we probe the local spectral properties of the rhodamine dye. Before any accumulation takes place, if you measure the spectra around the Janus particle, what you observe is the standard fluorescence spectrum of rhodamine. However, as soon as particles accumulate, what we observe is a nonlinear transition in the spectrum where both the line width and the intensity of this measured spectrum increases in a nonlinear way with the pump fluence, which is a typical indication of lasing. And of course, if you let the particle disperse again, what happens is that you reduce the local density of colloids, and so the scattering process, and therefore the peak decreases again, going towards the fluorescent background. And you can keep on playing like this. You can accumulate and disperse and move from the lasing to the non-lasing. So from the lasing to the fluorescence spectrum. Now, to make sure that what we observed was lasing, we did some other measurements, some of them already mentioned. We measured how the intensity of the spectrum and the line width of the spectrum changes as a function of the pump fluence, how much we're pumping our rhodamine dye. And in a laser, what you should observe is a nonlinear transition going towards a threshold, which indeed you can see once. So for the dots and for the diamonds, in the case of accumulation, you can see here there is this nonlinear transition where we set the lasing threshold. However, for the pre-accumulation case, you can see that these lines are pretty much proportional to the pump fluence, therefore just fluorescence. To support even further our observation, we also calculated analytically, we solved the transfer radiative equation for light as a function of the density of colloids and the critical radius of our system. And you can see here we can identify 3 regions: the red, the white, and the blue. The blue at low particle density and low critical radii is a region of no lasing. The red is a region of lasing for high particle density and high critical radius. And the narrow white line separating the two is the threshold when you change from no lasing to lasing. These are our experimental measurements. And again, you can see how they match decently well what we expected from theory. We have a no lasing region for low particle densities and small pump radius, so the size of our system. And we have lasing at the opposite end of the graph. And we have this white region where there is the crossing between the no lasing and the lasing regions. And now we can understand what's happening in the experiment for good. If we're starting from a situation, let's say if we decide our radius, so our pump radius, so the critical radius of the system, we are pretty much cutting a horizontal cross-section in this 2D surface plot. And if we're starting from a situation where the particles are dispersed, we are in the blue region where there is no lasing, so the line width and the intensity are more, they're broader and the intensity is lower, and this corresponds to fluorescence. But as soon as we increase the density of the colloidal particles around the Janus, we are pushing the system to cross the lensing threshold along this horizontal line until we end up in the lensing region, where we observe a smaller line width and a higher intensity for the lensing peak. And pretty much what we've done is to create a reconfigurable laser, where by dispersing and collecting the colloidal particle around the Janus, we can switch between lensing and no lensing at will. So now that we have seen that we can make this reconfigurable laser, let's see what we can do that cannot be done with conventional lasers. The first thing we can do is to create a laser switch in a macrofluidics sample. So if we combine two Janus particles, you can see at the top row here a schematic, and this is the experiment. If we have two Janus particles, and we start from a situation where the titanium colloids are dispersed, and we illuminate one or the other Janus particle selectively, what we're doing is to move the cloud of scatterers of titanium particles from one Janus to another and back. And we can do that iteratively by selecting each of the two Janus particles at one time. By doing this, we also observe a change in the spectral properties. At the beginning, the spectra are broad, like in the case of fluorescence, around each of the two Janus particles. But as soon as we collect the cloud of colloids around one particle or the other, you can see that we are generating lensing locally in one or the two spatial regions. And we can keep on switching between the two, of course. We can do more. We can get multiple Janus particles to work together to cooperate and create something that will not happen thanks to one Janus particle. For example, here we have a lower density of colloids, so the cluster around the Janus particle is not big enough to support lensing, but we can get multiple, in this case, two Janus particles to self-propel towards the original Janus colloid, bringing more titanium particle into the mix, increasing the size of the system and pushing the laser, the threshold of the system above the lensing threshold, and keep on narrowing it. And of course, we can do more fun stuff. We can do lensing geometries by having 2, 3, or 4 Janus particles placed in space. We can obtain a hourglass, a triangle, or a rectangle that lays. So going to the conclusions, what I've shown you today is that there are a lot of interesting cooperative and collective behavior that can emerge in the mix of active and passive particles. And what I'm suggesting here is that a lot of this behavior that haven't been explored yet are actually feedback behaviors in which both the active phase and the passive phase are interacting with each other in a feedback loop, modifying each other's behavior. And I'll show you 2 examples of this. The first one is the stigmage of active colloids, how the presence, the fact that the active colloids can create a field memory in the background of passive colloids can lead to increased cooperation by boosting group formation. And the second example I'll show you is a bit more applied, if you want, where the interaction of active particle and passive particles can give rise to nonlinear effects in the form of optical materials such as a laser. And finally, let me thank the people in my group that are all the ones that are actually doing the work. In particular, the work that I presented today was done by Manish, as I already mentioned. And Manish was really great at pushing the work through during the pandemic. Most of the work that I've shown you today, especially on the lensing side, was done in between openings between the national lockdowns. And of course, this work was done also with collaborators that I already introduced. In particular, the first part, the stigmergy active colloids, was done in collaboration with Nuno and Cristobal at the University of Lisbon. And the work on the laser was done with Ricardo, Druve, and Kit at Imperial College London. This work would not be possible thanks to funding, of course, and I would like to thank you for inviting me here, hosting me here, and for your attention.
if there is any question, please feel free to ask. The floor is open.
So, Giorgio, what if the particles are itself coated with dye molecules instead of the entire medium? Would that still show lacing?
In principle, yes. So there are more than, I guess, more than coated. You could use particles that contain dye. I guess in the case of dye, you would probably need to reach a certain density of dye in the particle to achieve absorption and drive the same effect. But in principle, I would think so. So the things that you need to strike a balance between absorption to drive the mechanism, but you don't want to have too much absorption because absorption introduces losses, and losses can be detrimental for lacing. So in principle, I guess anything that absorbs, if done properly, can lead to the same behavior.
Okay, thank you.
So if there is no other question, then I can ask. Like, sir, you have shown in this lacing work that you have used genus particle, and you mentioned that instead of genus particle, even if a absorbing colloid is being used, then also it will serve the same purpose. So what is the, because in case of genus particle, the temperature distribution will be asymmetric. So is there any role of that?
You mean the distribution of the heating?
Yeah.
Yes, in principle, there could be some effect. I mean, what we observe is that like 90% of the times, once the cluster is formed around a Janus particle, the Janus particle tends to orient facing down.
Yeah.
So in a sense, that asymmetry that should be there is kind of compensated by the fact that of this reorientation dynamics. And the reason for that is that once the Janus particle is blocked by all the big cluster of colloids, it cannot self-propel anymore. Therefore, the same align and the carbon cap side is heavier, right? It's denser. So yes. Once you kill the self-propulsion, then there are no more aligning interactions to the substrate. The standard rotational dynamics prevail, and that tends to realign them with the cap facing down. So, but to be honest, I mean, obviously you will be creating an asymmetric temperature profile, but I don't think that will change particularly what we observe because of the R² dependence. Yeah. You know, even if there is a bit of an asymmetry, you will still observe similar behaviors. Okay. I think.
Okay. And in this line, there is one more question that, okay, if here the genus particle are floating, like it is not stuck, so what if it is stuck to the glass surface?
Yeah, I mean, I think you should still observe the same behavior. Like, imagine that drop the Janus particle, the colloidal particle. Yeah. What you could do is to have a disk of evaporated material, gold or carbon, that is stuck on the substrate. That will still create thermosmotic flows, right, around it. Yeah. It just, you lose the possibility of moving the heat center, the local heat source, but the accumulation will still happen. And in fact, I think also, Pavan, you also done some stuff with the gold on surfaces, right? I know Frank for sure. What Frank does in Leipzig, usually they cover their glass substrate with gold, and then they use that to generate heating. So, you know, it would work too. Again, the question is whether what you're using to create this heating is introducing enough absorption to drive the mechanism, but not too much to kill the lensing effect because of the losses.
Okay. And from the last part of the talk, I have one more question, because you have shown the self-propulsion of genus particle, and the another particle will follow the same path. But the self-propulsion of genus particle is also there when there is no phase transition in the medium. Solely depending on the temperature field, they will self-propel. So what is will you observe similar collective motion in that case?
Yes, sorry, maybe I wasn't too clear about that. So the critical mixture, we only use it in the first experiment I presented. In that case, you have a phase transition. In this case, what we have been working with is just as ethanol. So there is no, the mechanism for propulsion is different. It's thermophoretic. It's not a critical demixing.
Yeah, okay. But in the previous case also, the last work that you have presented, so there, because you have shown the self-propulsion and the interaction of the path tracing or by another genus colloid, okay, in the same path. But will you expect the similar kind of behavior in case of when there is no water lutein mixture, only water is there?
For the first experiment, you mean?
Yes. Because self-propulsion of genus particle will also be there in that case because of the thermal asymmetry.
It's a good question. I'm not sure I would expect the same, because I think that you will miss the torque mechanism.
Okay.
I think. I mean, what will happen in just water because of thermophoresis? But first of all, the temperature that you need, practically, experimentally thinking, the temperature that you will raise to get the Janus particle to propel will be much higher than in the critical mixture, which will also drive probably some thermo-osmotic flows and move the passive obstacles. But we haven't done the experiment. So actually, that's not true. We have done the same experiment for the lensing in water rather than in ethanol, and we observe accumulation of colloids around the Janus particle. So I think that the difference, the main difference is that in the experiment with the water lutein, the particles, the Janus particle, because of the critical demixing being asymmetric near the obstacles, they tend to steer away from the obstacles, so they tend to avoid them. In the case of Janus particle in water, you will probably increase the chances for collisions, so you will not observe the same field memory emerging.
Okay. And sir, one more question. Like in the previous case where you have shown the active-passive mixture in lutein, water lutein, active-passive particles in water lutein mixture. So there is there any size dependence between these passive colloids? Like you have shown the dependence of the percentage of the passive colloids in the system, but will there be any kind of size dependence?
Yeah, I'm sure. I'm sure. So in our case, the size of the passive colloids is the same as the Janus, and we stuck to that ratio because we wanted to keep the configuration 2D mainly. We tried with slightly bigger passive particles. So in our case, in the experiments that I've shown you, we are 5 micron for both. We tried with 7 micron passive and 5 micron active, so we still had a pretty much 2D geometry. We saw similar effect. Obviously, you are changing, by changing the size, you're not just changing the size, you're also changing the weight of the particles, right? So then the transient path, the form will follow different dynamics. There will be probably, if you go to bigger particle, they will relax on a longer time scale. If you go to smaller particle, they will relax quickly. And that might affect quantitatively the emergence of the behavior, but I don't think it will affect it qualitatively because we tried that in simulations for a bigger range of systems. In experiments, we were limited by what we could do by keeping the 2D geometry. And we observe pretty much the same behavior, just shifted in time or space. So I think it's quite robust, but we haven't done many experiments beyond the 2D configuration. So, everything is based on what we done with simulations to double check. Yeah, so the experimental proof might contradict me in the end, but I would think that the behavior should be robust.
So, sir, there are few questions in the chat also. So, like, what is the cause? You can check.
Yeah, so the torque, let me go back to slides. So this is like a macroscopy image of the mixing around the Janus particle. I think the yellow here is the water rich phase, and the orange is the lutein rich phase. If you don't have any obstacles, the mixing is symmetric along the axis of the direction of propagation of the particles. If you now place an obstacle, let's say here, on one side of the Janus particle, you will be preventing the mixing from happening on this side, but it will still happen on this side. So you will get an asymmetric demixing from one side or the other of the Janus particle. So in this case, for this configuration, the particle will rotate towards the region of higher demixing. So in this configuration, it will be counterclockwise. But if you take the opposite situation where the obstacle is on the other side, then the demixing will be larger at the top rather than the bottom, and then you will rotate clockwise. Basically, whether it rotates clockwise or anti-clockwise will depend on which side of the Janus particle sees the stronger demixing, so away from the obstacles.
So, sir, from the same line, from the same slide, I have a question. Like here, the lutein rich phase and water rich phase shows different color. Have you treated your sample in some different way?
Yeah, so in these experiments, we added some dyes. I think they were rhodamine dyes, just to visualize the because rhodamine is not very hydrophilic, right? Doesn't like water that much, so it will prefer to stay in the lutein rich phase. You can see that where the system is not the mix, you get like this orangey, because obviously there is rhodamine, but as soon as you get the mixing between the water and the lutein rich phases, the rhodamine will primarily be in the lutein area, and therefore that's why you see the change in color. I think there was another question in the chat: what happened to the torque and path following effect if we use active droplets in place of coated Janus particles? It's a good question. I have no idea. I don't know, because I think some active droplets also show some sort of self-repelling behavior. I think there is work from Corina Mas that has shown that, but I honestly don't know. It would be an interesting experiment to try. I no idea. I'm not too familiar with active droplets. I think a lot of the mechanisms that they use are driven by the formation of mast cells. So I guess that could be something that could be influenced by the presence of obstacles, because it's also some sort of phase separation behavior. But then whether you will end up observing something similar or not, I really don't know. It would be a really interesting experiment to try, though. Yeah, so sorry, I don't have an answer for the question, basically. How long these Janus particles move and what is the time duration of motion? As long as you keep them going, as long as you keep providing them with light, they will be happy and they will keep on moving. I think our experiments were done in half an hour. All of the experiments that I've shown you, we stopped after half an hour. not mainly because most of the Janus particle that we started with would have joined a cluster. So at that point, I think after 25 minutes, particle will mainly be in clusters.
Okay. So is there any more question?
Does the size of Janus particle have any effect? Probably. Again, so if you change the size of the Janus particle, you're changing the persistent length. So how quickly they will go back to diffusive motion. So that will also change the dynamic. I would expect that to be a change, not a qualitative change, rather a quantitative change. There is one problem that at least we found in the lab. If you want to make too big Janus particles, they're not gonna self-propel. And we think it's because the coating is too, then it doesn't come out as a film. But I think once we went above 7-8 micron in size, I mean, they didn't go anywhere. So I don't know. Obviously, in experiments, you have a bit of variability. You can probably go from 1 micron to 6-7 micron, and that's about it. And there are a lot of questions. Shall I keep on answering?
Yeah, sir. Please.
So what is the typical thickness of carbon coating? So for these experiments, we had 16 nanometers. And we use a carbon spatterer to coat them. There are works that have used, you don't need as much, you need probably 10, 20 nanometers of carbon are more than enough for Janus particle to move. And yes, thickness will have some effect on motion because it will increase the efficiency of the light absorption. The more material you have, and up to a certain point, because I think this light absorption effect, they are more like skin depth rather than bulk effects. So once you go above a certain thickness, which is probably a few tens of nanometers, you will just you will probably become detrimental to the motion because you have a very dense, heavy layer. Another thing to keep in mind is that with carbon, if you grow it too much, it will probably detach from the Janus particles too. So, you know, what will work is anything between 10 nanometers to 100 nanometers.
So, okay, this is if this is done.
So hopefully, Giorgio, we will be having you sometime here in Pune.
That would be nice, but I think we also need to continue the chat we started in Japan, right? And I was looking for whether there is, I think there are some schemes in India and the UK. They're just not open yet, but I've been looking for.
Yeah,
anyway.
I've been also tracking some of the things. Yeah, I'll get back as soon as I have some more information.
Thank you for having me online.
Yeah, so thank you, sir, for giving such a wonderful talk. We hope to have you here in Pune at some point of time. Thanks again.
That would be nice.
Yeah, thank you, sir.
Thank you very much. It was nice meeting you all. Thanks.
Bye-bye.
Bye. Bye, Pawan.